This is lecture 8C. It's continuing with renal physiology, and it's on acid-base balance in the renal system. Now, acids, of course, acids are proton donors, and bases are, and it's a little bit vague to say they're proton donors and bases are proton acceptors, but that's what they are. The normal pH in body fluids varies depending upon which part of the body you look at. If you look in the arterial blood, the normal pH in the blood plasma is about 7.4 with a range of plus or minus 0.05. So the normal range would be 735 to 745. On the venous side and in interstitial fluids, in almost all parts of the body, it's a little bit lower. It's 7.35, which, maybe you should think about why that might be. Intracellular fluids are at a pH of 7. Now, alkalosis and acidosis, also called alkalemia and acidemia, but more commonly called alkalosis and acidosis, refer to changes in the condition of the arterial blood when the pH is above 7.45 or below 7.35. Now, a great deal of the importance of the maintenance of acid-base balance relates to the changes in shape of proteins that occurs at when the pH in their environment changes. Now, 99.99% of enzymes are proteins, and shape or structure determines their function, and therefore any shape change can radically cause them to have a shift in their function from functional state to decreasing enzymatic activity. And so maintenance of shape is vital, and therefore maintaining a proper pH is also vital to that. As you should remember, you learn this, but maybe you never really place it into context and you kind of happily forget it, but as you should remember, the reaction rate of an enzymatically catalyzed reaction really has a typical normal distribution around an optimal pH. As the pH shifts away from the optimal level, the enzymatic activity will drop until it actually can get all the way down to 0 when you have such large shape change in the enzyme that it is no longer can no longer interact with the substrate and is therefore no longer functional in terms of being a catalyst. Now, you should, of course, be able to appreciate the fact that when you do look at a range of pHs versus the catalytic activity or enzymatic activity or ability to produce product, all the same thing. If you look at a different enzyme, if you're looking at comparing two different enzymes, they may have quite different pH profiles in terms of their activity. Here you see one where the optimum pH is at about 6 in terms of its activity, and another one here where the optimal pH is somewhere just above 10, I'd say, or at 10. So that's quite a big difference. Depending upon which enzyme you're looking at, they can have quite different optimal pHs for their activity. And this is shown in these three examples here. I'll first get rid of these lines that I drew. Here's a relative reaction rate or enzyme activity for three different enzymes. Pepsin, which you might remember if you learned about it. I'm not sure. I don't know if you learn about this in this enzyme in general biology too, and normally in this course we would have covered it for digestion. Pepsin is a protease enzyme found in the stomach, secreted as an inactive enzyme called pepsinogen, and once it gets into the acid environment of the stomach, it becomes activated. And I pretty much gave away why its optimal activity is found at pH 2, because it's in the stomach and it's an acid environment, and therefore it can break down proteins, which is what the enzyme activity has. It can break down proteins optimally in an acid environment. On the other hand, salivary amylase, enzyme produced by the salivary glands, which can break down starches, has its optimal activity at pH just below 7, which is what you would find, in fact, in the mouth as you eat. And so that's where its optimal activity is. But there are other enzymes that have their activity at an even higher alkaline pH. Here's arginase, which is a key enzyme, the final enzyme in the biosynthetic pathway leading from, it catalyzes the conversion of ammonia to urea. Ammonia is derived from nitrogen waste, metabolic waste, from the amino groups in amino acids that get digested. So here you see the clear evidence that you need to maintain particular ideal pHs, that they vary in different places. Most of the pH, if intracellular typically is 7, then most of the enzymes acting inside would have an optimal pH of 7. If the pH is 7.4 in arterial blood, then those enzymes that are found there would have, depends on the area, but it's all maintained in a balance, and it's very tightly controlled. If you looked at you should put this into the context also of pathways, metabolic pathways involving a number of different enzymes. Here you see on the left the glycolytic pathway leading from glucose down to production of pyruvate. This is a 10-step metabolic pathway converting glucose into 2 pyruvates, and each step is catalyzed by a unique enzyme, and some enzymes are more pH sensitive. In other words, they have a much narrower optimal range of activity than, say, a different one, which might stretch out quite a bit wider. The most sensitive enzyme in terms of being pH sensitive in this pathway of glycolysis is phosphofructokinase, which phosphorylates fructose at the 1 carbon position. So if this enzyme here in this pathway is inhibited, then the pathway will stop. It'll shut down. The substrates will build, these substrates for that enzyme will build up in concentration. None of the final product, pyruvate, will be formed if this is outside of its optimal, far outside of its optimal pH range. If it's a little bit outside, then the whole pathway will just slow down. But again, you can see the importance for metabolic pathways in terms of pH. The metabolism of the original substrate, in this case glucose here, is dependent upon the weakest link in the chain in terms of pH, the most pH sensitive enzyme, in this case phosphofructokinase. Hopefully now you've got the bigger picture and understand why acid-base balance is such an incredibly important homeostatically regulated physiological mechanism in the body. Let's move on. Okay, in terms of sources of hydrogen ions in the body, most originate from cellular metabolic activity, particularly the breakdown of phosphorus-containing proteins. And I can tell you a very high percentage of proteins in the body are phosphorylated, and this results in the release of phosphoric acid into the extracellular fluid. Another source of hydrogen ions is the anaerobic respiration of glucose during lactic acid fermentation, which produces this acid, of course, lactic acid. Also, fat metabolism, breakdown of fats, can generate ketones, which are quite acidic. And of course, as we well know, transport of carbon dioxide, which is continually being produced from the metabolic breakdown of carbohydrates in cells. The transport of carbon dioxide occurs in the blood as bicarb, and that also results in the release of hydrogen ions. Well, hydrogen ion regulation can be regulated, the concentration of hydrogen ions in the body, and of course this is what determines pH. So pH is related to concentration of hydrogen ions. It is regulated by the chemical buffering systems in the body, which can act within seconds very quickly to buffer or reduce any major shift in pH. By also the respiratory center, which is in the brain stem, and that can act within minutes to trigger a response in respiration that can mediate control of pH, and by the renal mechanisms in the kidneys, which require hours to days to take effect to make a change in pH, but in fact are probably the most effective way, or more effective at least, than the respiratory mechanism of regulation of pH. So in terms of chemical, we'll go through those three. In terms of chemical, and the last two, by the way, are physiological mechanisms, whereas what we're talking about now is a chemical buffering system. I remind you that strong acids, the reason they're called strong acids is because all the hydrogen ions associated with them will completely dissociate free when these compounds are in solution in water. Weak acids, on the other hand, will only partially dissociate their hydrogen ions, and therefore are pretty good, if they're present, are pretty good at preventing major shifts in pH. Similarly, strong bases will easily dissociate in water, quickly tying up hydrogen ions. They'll interact with hydrogen ions. However, weak bases will accept hydrogen ions more slowly, and these include bicarbonate, sodium bicarbonate, and ammonia. These buffers act to resist pH changes when a strong acid or a strong base is present in a solution, or in the lab added to a solution. And all of you, I presume, have done titrations using NaOH or HCl. So you have some familiarity with that. There are three chemical buffering systems in the body. The first important one is the bicarbonate buffering system, which you are somewhat familiar with, the phosphate buffering system, and the protein buffering system. Any drifts that occur in pH are immediately resisted by all of these chemical buffering systems, whichever one are present in the location where the pH changes occur. So let's go through these. The bicarbonate buffering system involves carbon dioxide and water and carbonic acid and sodium bicarb. So you have this mixture of carbonic acid, the salt, sodium bicarb. And you are familiar, of course, with the idea that carbonic acid, which is a weak acid, will form when you have CO2 and water if you have carbonic anhydrase present, and then that carbonic acid will dissociate into hydrogen ions and bicarbonate. And the bicarbonate also is a buffer. The carbonic acid dissociates into bicarbonate and releasing some hydrogen ions, but the hydrogen ions don't have that much of an effect because they can still combine with form carbonic acid. If there's too many of them, it'll go the other way. So this is the bicarbonate buffering system, a very important system in the blood plasma. If a strong base is added, it reacts with the carbonic acid to form sodium bicarb. pH of the solution will not rise tremendously because of that interaction. The bicarbonate buffering system is the only important extracellular fluid buffer. Okay, that's the only important extracellular fluid buffer. The phosphate buffering system is important in intracellular fluids, also in the urine. So the phosphate buffering system is similar to the bicarbonate system in that you have a weak acid, which is dihydrogen phosphate, H2PO4-, and a weak base, which is monohydrogen phosphate. So you have dihydrogen phosphate or monohydrogen phosphate, and they can go back and forth depending on the situation. This weak base can become this weak, oh dear, I made a mistake. This weak base can become a weak acid if there's an accumulation of hydrogen ions, and vice versa. It can shift back and forth, nicely buffering the intracellular fluid and also urine. The third chemical buffering system are the proteins. There are lots of plasma proteins. This is extracellular in the intravascular fluids. There are also intracellular proteins. So this is extracellular, and this is intracellular compartment. These are the most powerful, most plentiful buffers. They're all over the place in the body. And you should realize, of course, that the reason is that some of the 20 amino acids have a free acid group. They can act as weak acids, and some can act as weak bases. In fact, some of them have an amino group and an organic acid group attached to the central carbon atom, and therefore are amphoteric molecules. And so they will be able to act as both weak bases and weak acids depending upon which group is interacting with the acid or base. Many proteins, in fact, are amphoteric molecules. Every amino acid, in fact, is also. When you think about it, a free amino acid will have an amino group and a carboxy group attached, one acting as a weak base and or acid, and the other one acting as a weak base. Okay, let's move on to strict physiology, and that is the two physiological buffering systems that act in the body to maintain homeostatic regulation or balanced pH. They are the respiratory system and the renal system. The respiratory system can regulate acid-base balance. It's a physiological buffering system. And the reason is because of the reversible equilibrium that you're familiar with between, within this equation. It can reverse back and forth depending upon the situation, and in that way it can have a buffering effect on the pH level. I'll explain that more. You have dissolved CO2 in the blood plasma, and of course you have water. It's a water-based solution. These can interact inside red cells to form carbonic anhydrase. Did I just say carbonic anhydrase? to form carbonic acid, and this can be mediated by the enzyme carbonic anhydrase. Carbonic acid will, in response, split into hydrogen ions and bicarb, and the bicarb will leave the cell and go into the plasma. This is typically on the venous, will be on the venous side of the circulation. So you have all of these players, and it can shift back and forth depending upon the balance within the equation. And I'll tell you what I mean, so hopefully you'll grasp what I mean by the balance, how the respiratory system can shift the balance so that it goes in one direction or the other. Okay, if you are unloading carbon dioxide, in other words, breathing it off, then the hydrogen ions will bind to bicarb as the bicarb moves into the red cells and forming carbonic acid and shifting over. The whole reaction here will shift over in this direction as the carbon dioxide disappears, because the person is blowing it out every time they expire or breathe out. If they breathe out faster, they will unload more carbon dioxide, and the equation will shift even more from right to left. So the respiratory system, of course, could shift things either way, in fact, and that's shown here. Well, let's go through a few basics. Of course, this reaction can go in either direction, from right to left or from left to right. It depends on what is built up more or in more concentrations. If the PCO2 is high in the body, then that's hypercapnia, high CO2 level, and that will lead to acidosis because the reaction is driven to the right. If you have more of CO2, then the reaction will get driven in the right direction, from left to right. And as a result, you'll have a rise in the hydrogen ion concentration, and you can get acidosis. In other words, the pH drops. Now, the response to hypercapnia, or unusually high levels of CO2, is deeper, more rapid breathing. And people will do this unconsciously. They will not be aware of it, but their brainstem will respond and trigger trying to dump more CO2 and get the reaction. As a result of the getting rid of more CO2, the reaction should go from right to left. And so you get rid of hydrogen ions and the pH goes up. Okay? That would move the reaction towards the left. Hydrogen ion concentration would be reduced and pH would rise back into the normal range if the person breathes faster, deeper and faster. Alternatively, if a person is not in acidosis but alkalosis, if their pH is 7.45 or above, then that rise in rising plasma, no, that's wrong. Okay, let's cut that out. Okay, so alkalosis is when the pH is higher, and the respiratory response is to slow down. The brainstem will sense that and get that information also from chemoreceptors in the aortic body and the carotid bodies, and you'll get slower, shallower breathing, resulting in the reaction up here moving from left to right as the CO2 builds up in concentration. So here, what you have is an increase in hydrogen ion concentration and a decrease in CO2. I'm sorry. What you have here is because the person is breathing slower and shallower, you have an increased level of CO2, which drives the reaction more to the left, causing a rise in hydrogen ion concentration, and that causes the pH to drop. That's the respiratory oh dear. Yes, that's the respiratory it's complicated. That's the respiratory response to alkalosis, a high pH. Slower, shallower breathing, resulting in more CO2 building up in the body, less of it's getting blown off because of the slower, shallower breathing, resulting in a shift towards the right side, a rise in hydrogen ion concentration, and therefore a drop in the pH back to within the normal range. Yeah, that's out. Okay, that's the respiratory response to shifts outside of the normal range of pH, alkalosis or acidosis. Let's talk about the renal system. Now, chemical buffers in the body can tie up a lot of excess acids and base. However, the only way to permanently eliminate them from the body, which will permanently solve a problem pH balance, is through the renal system. The lungs can eliminate carbonic acid by blowing off carbon dioxide, but they cannot sustain that over a long period of time. These people cannot, for example, try and eliminate carbon dioxide all the time if they have some sort of metabolic acidosis. They're trying to get rid of that increased carbon dioxide, blowing it off, so they're breathing, they're huffing and puffing, they're breathing as much as they can. It doesn't work. It can't be sustained. Only the kidneys, only the renal system can rid the body of metabolic acids like these, phosphoric acid, uric acid, and lactic acid, or ketones building up, by eliminating it, and in that way preventing acidosis. Okay, so the ultimate acid-base regulatory organs are in fact the kidneys. So we're going to spend quite a bit more time looking at renal mechanisms of acid-base balance, which is why this topic is covered in renal physiology. Now, hydrogen ion secretion does occur. It can occur in the proximal convoluted tubules, and it can occur in the collecting ducts. And it leads, of course, if you have increased secretion of hydrogen ions, it'll lead the pH to rise back up into a normal range. Hydrogen ions often come from the dissociation of carbonic acid in this equation. This is, of course, the key equation that we've seen before. So hydrogen ions are coming from the dissociation of carbonic acid into bicarbonate and hydrogen ions. The most important renal mechanisms for regulating acid-base balance are these three. They involve reabsorption of bicarbonate ions. This is happening in the kidney, reabsorption, or generation of a new bicarbonate, or excretion of bicarb. And we'll deal with these each individually. There's actually two ways of generating new bicarb. Two mechanisms that can generate new bicarb, and I'll cover those. So, reabsorption, generation of a new one, or excretion of bicarb. You should realize that the first two, reabsorption or generating a new bicarb, so that goes into the body, is equivalent to losing a hydrogen ion. The effect in terms of the body's pH balance is that it's equal, that these two are equal to losing a hydrogen ion, and therefore cause the pH to rise. Similarly, although opposite, but similarly, excreting a bicarb, the third mechanism, excreting a bicarb, is the same as gaining hydrogen ion, and therefore that would cause the pH to drop and move back down into the normal range. So if a person has excess hydrogen ions, they can reabsorb bicarb or generate new bicarb. If a person has a deficit of hydrogen ions, a bicarb can be excreted, and that's equivalent to gaining a hydrogen ion and causing the pH to drop. We're going to go through these one at a time. Let's look at reabsorption of bicarb. Okay. And all of these things are happening within the tubular system of the kidneys. This equation, of course, is key. Carbon dioxide combining with water to form carbonic acid, dissociating to hydrogen ions and bicarb. And this is what happens is that you have bicarbonate getting reabsorbed by tubular cells in the proximal convoluted tubule as a hydrogen ion gets secreted. So each time a hydrogen ion gets secreted into the tubular system, and this is all part of the secretion. I told you about the three mechanisms that are occurring in the kidney: filtration, reabsorption, secretion. This is one of the things that can potentially be secreted. Hydrogen ions secreted, and a sodium and a bicarb ion get reabsorbed, resulting in a rise in the pH. The secreted hydrogen ions will form in the tubular space, in that fluid, will form carbonic acid, and therefore what you get is bicarb disappearing from the filtrate at the exact same time as bicarb enters the peritubular capillary blood. And that may not be obvious, but should be clear from the next diagram. And the steps are shown here. Each of these steps that are numbered in this diagram: 1, 2, 3, 4, 5, and 3 here. Ca stands for the enzyme carbonic anhydrase, which you need to convert CO2 and H2O into carbonic acid, and that can occur inside the cell. This is a cell lining right here is the outline. I'm going to draw it differently. These are cells that are these are cells that are lining the tubular space. So this is the outline of the proximal convoluted tubule cell lining the space. Here's the next one over. Okay, and here another one, and this is the space here of inside the tubule, and here are the peritubular capillaries on the right. They're actually very close overlying. So here you have the peritubular capillary. This is the proximal convoluted tubule cell, the cell lining the tubule, and the filtrate in the tubulum. And each of these steps is outlined here. Remember, we're looking at the reabsorption of bicarbonate, which is physiologically the equivalent, and it is happening, as you can see, the equivalent to losing a hydrogen ion. So how this occurs is that inside these cells in the proximal convoluted tubule, you have formation from CO2 and water. You have formation of carbonic acid, which dissociates into bicarb that gets reabsorbed, cross into the body, into the bloodstream. and hydrogen ion is being secreted out, and then the hydrogen ion is combining with carbonic acid, bicarbonate to form carbonic acid, and that splits under the effect of this enzyme carbonic anhydrase to water and some dissolved CO2, and is excreted out in the urine. So at the same time as a bicarb molecule is being produced here in the tubular fluid, a bicarb molecule is reabsorbed into the body. So that's reabsorption of a bicarbonate molecule and concomitant secretion out of the hydrogen ion here. And that's gone through in each of these steps labeled 1 to 6. It's all described there. Okay, so that was reabsorption of bicarb. Now we'll talk about another mechanism, generation of a new bicarbonate ion, same as losing hydrogen ion. There are two mechanisms that can carry this out within the collecting ducts, actually. They can generate new bicarbonate ions. They involve excretion of acid via secretion and excretion mechanisms, but the acid is hydrogen ions or ammonium ions are being excreted out. They're lost, and therefore the pH will rise as a result of that. So this is generating new bicarb to control pH balance in the body. Hydrogen ion excretion involves the hydrogen ions. You're taking in hydrogen ions in the diet, and that intake has to be counteracted by generating new bicarb molecules. They need to be excreted out and they bind to buffers in the urine. I wouldn't worry about which cell does this, but it's in the collecting ducts that secretion or excretion of hydrogen ion occurs, and it gets buffered and excreted out. The new bicarb that is generated through this will move into the interstitial space and then into the peritubular capillaries. So here you see generation of new bicarb by involving hydrogen ion excretion. This would be in response to acidosis. In a condition where there is acidosis, what you get is production of a new bicarbonate here, and it's going in to the bloodstream. At the same time, the hydrogen ion is being secreted out into the urine, combining with phosphate, monohydrogen phosphate to form dihydrogen phosphate, and goes out in the urine. This is not the same mechanism as before. It involves formation of the pathway going from water and carbon dioxide towards forming a new bicarb and reabsorbing that in, or absorbing that in. So you have generation of new bicarb by hydrogen ions excretion going out, and generation of a new bicarb that goes in. The other type of generation of new bicarb involved ammonium ion excretion. So the ammonium ions are produced by the metabolic breakdown of the amino acid glutamine. Okay, so that amino acid is metabolized producing ammonium ions and bicarb ions, and the bicarbonate that's generated, a new bicarb these new bicarb molecules that are generated will move into the blood, and the ammonium ions, which are acid, will excrete into the urine. And here you see that coming from glutamine, going to bicarb that gets taken into the body. So a new bicarb is taken in, and two actually per glutamine, and the NH4 is moved out into the tubular space and will go out into the urine. The ammonium ion will move out and go into the urine and be excreted. So this is a second way. I should point out that all of these things will cause a rise in pH because you're getting rid of acid. So this is a way that the body can get rid of acid using the kidneys to mediate this. I think for the third one I think I'll stop here, and this will be part 1 for Unit 8C, and we'll do the rest as a separate part 2 for Unit 8C.